Hi, I am Brinda Rana, and I'm a genomics researcher at the UC San Diego Center for Healthy Aging. As a genomics researcher, I study DNA, but I also study the milieu of molecules in our body, including RNA, which are responsible for regulating the activity of our genes, metabolites, which are the product of metabolism in our body, and proteins, which are the product of our genes. I study how our day-to-day lifestyle and changes in our lifestyle influences these molecules, and how our physical environment also influences these molecules. Our environment includes exposures such as pollution, stress, pathogens, and even the coronavirus. Recently, researchers around the world have become interested in small vesicles called extracellular vesicles that are shed by cells in our body and contain molecules that represent the cells from which they were shed. These extracellular vesicles represent molecules contained in different tissues in our body. Circulating extracellular vesicles may be a way in which different tissues talk with each other. We can isolate these extracellular vesicles from blood samples. Studies on extracellular vesicles, or circulating in our blood, provide clues about what cells and tissues in our body are doing and how they are affected by lifestyle and the environment. We are particularly interested in the function of endothelial cells. Endothelial cells are the cells that line the inside of our blood vessels and ultimately determine the health of our blood vessels. Because our blood vessels are important in supplying nutrients to all our tissues, maintaining the health of these cells as we age is important for our overall health. Endothelial dysfunction is predictive of heart disease, as well as many other diseases. In fact, it's been proposed that a mechanism for the observed COVID-19 related cardiovascular comorbidities that are seen in older adults may be through vascular endothelial dysfunction. My collaborators, Dr. Sears and Chang, have developed a methodology for isolating extracellular vesicles that are shed specifically by endothelial cells, and for characterizing small RNA molecules called microRNAs within these particular extracellular vesicles. They have demonstrated that lifestyle modification, in particular less sitting, is associated with changes in the microRNA content of endothelial cells, extracellular vesicles that are circulating in the blood. While the pandemic temporarily closed down many of our ongoing studies, it has opened new research questions that are important to address for immediate concerns in future pandemics. One of the research questions we're interested in is whether we can apply this new method to identify novel biomarkers in order to predict or monitor the risk of endothelial dysfunction-related cardiovascular disease in older adults with COVID-19. Another question of interest is, has the change in physical activity due to the COVID-19 pandemic affected our endothelial cell health? The pandemic has definitely affected the way we work in our laboratories, but we continue to adapt our research to address the health issues our community faces today.